Hello, everyone. Thank you for joining us for part one of Azenta Life Sciences Advancing Transcriptomics, Gene Expression Screening, Single-Cell RNA Seq, and Beyond webinar series. My name is Brianna King, and I'll be moderating today's event alongside my colleague, Sibi Sabu. Today's webinar will be led by Dr. Andrea O'Hara, who is a Strategic Technical Specialist within Azenta's NGS team and has more than 11 years of experience with NGS. She earned her Ph.D. from the University of North Carolina at Chapel Hill in genetics and molecular biology. There's just one thing I wanted to point out before we get started. Throughout the event, please feel free to send any questions you have through the Q&A chat box located on the right-hand side of your screen. Dr. O'Hara and our Q&A panelists will address your questions during the Q&A portion of the webinar. Without further ado, I'll hand everything over to Dr. O'Hara to begin the presentation.
Thanks, Brianna, for that great introduction. So today I'll be presenting to you Advancing Transcriptomics, and we'll be covering a variety of different topics, including gene expression screening, single-cell RNA-seq, and more. Just to get started, as a brief recap, you may already know us as Azenta, but if you haven't heard of Azenta before, we're actually a union of a number of different companies, and we've rebranded under one name. So you may know us previously as GeneWiz or Brooks Life Sciences. As you can see here, there's actually a number of different companies within this unit. However, again, all rebranded to be called Azenta. Now, at Azenta, we actually offer a wide variety of different products and services. Today, I'll be focusing specifically on our genomics and analytical services, but we do also offer sample sourcing, storage, automation, and logistics, consumables and instruments, data and informatics such as LIMS systems, and then consultative services. And the consultative services actually covers everything you see here, including things like what we're doing today. So as I mentioned, today we're going to focus on the genomics and analytical services. We have sites located across the globe. Our main sites for the genomics and analytics are going to be in New Jersey in the United States, in Leipzig, Germany, and then in Suzhou, China. Now, today we're going to be focusing specifically on transcriptomics. And when we think about transcriptomics, we have a few different tools in our toolkit. I have our four main highlights here as shown. So we're going to be discussing each of these in more detail. This is going to include Illumina-based RNA sequencing, which is short-read sequencing, PacBio IsoSeq, which is long-read sequencing, 10x Genomics, which is a single-cell RNA sequencing, and then NanoString Genomics digital spatial profiling, and this is spatial RNA sequencing. I'll also be touching on some of our proteomic solutions, which can be used as a downstream way to validate the results that you found. So let's get started with our bulk RNA sequencing methods. To get started, I want us to think about a fruit salad. We're going to come back to this metaphor a few times. But when we're thinking about a complex tissue, think of a fruit salad. We're going to have a lot of different cell types or different pieces of fruits. And we can have more than one kind of tissue or fruit salad. We could have a fruit salad that's very heavy in apples and bananas. We could have a fruit salad that's really heavy in different kinds of melons. When we look at our bulk RNA sequencing, we're taking that complex tissue or that fruit salad, and we're creating a fruit smoothie. And that means that when we're tasting our fruit smoothie or sampling our bulk RNA sequencing, we're going to get a good idea for the frequencies of the types of fruits or cell types that are present, but we might not get all of the nuance. So for example, I will be able to tell if I have a fruit salad that was really heavy in strawberries and bananas in my fruit smoothie, but I might not be able to taste it if there was only a single apple slice that was in there. So that bulk RNA sequencing is really, again, our fruit smoothie approach, where we're going to get a really good view of the overall flavor profile. We might get some undernotes, but we're not going to be able to detect everything in huge detail. This is great for us, though, to get a really good overall view of what's happening in any particular sample. So for Illumina-based RNA sequencing, this is a bulk RNA sequencing approach. And we have a few different flavors or types that we're going to choose from. So we can start with poly-A selection. That's where we're going to take the poly-A tail, and we're going to get everything that is polyadenylated. We can use an rRNA depletion or total RNA approach, which is going to allow us to see more. Or we can do small RNA sequencing, and these are really meant for our microRNAs, which have special modifications. I'm going to focus in on the poly-A and rRNA in just a little more detail. So if we look at all of the different kinds of RNA sequencing that we have in our bulk sequencing approaches, we have four main tools at our disposal: high throughput gene expression screening, mRNA sequencing, total RNA sequencing, also known as rRNA depletion, and iso-seq. I want to focus on just these middle two to get started. So again, with the mRNA sequencing, we're taking everything that has a poly A tail and pulling that out. So we're looking only at polyadenylated RNA. This means we're really only looking at RNAs that are going to be turned into a protein. When we use this type of approach, again, we're doing a selection-based method. We need great high-quality RNA because we need to be able to have that whole transcript intact to pull it apart. We're going to want to target approximately 10 to 30 million reads for most studies. 20 million is most common. And we'll be able to get different kinds of information from this. So in addition to getting, again, all of those RNAs that are going to be polyadenylated and turned into a protein, we'll get our gene expression from that. We'll be able to detect variants because we've pulled out that whole transcript. We'll be able to tell alternative splicing. We'll be able to say if there's multiple transcripts, which one we have presence, and we can use this data for transcriptome assembly. You can see here that we think of cost. This is going to be a mid-range cost. Total RNA sequencing is going to take everything that's in this pool, that it's going to include all of those poly A RNAs, but as well as things like long non-coding RNAs, transfer RNAs, and it starts out with ribosomal RNA in there as well. Now, ribosomal RNA makes up so much of that pool that we'll do an rRNA depletion or ribosomal RNA depletion to remove as much of that as we can. And then we're going to move on with our sequencing. Now, when we do this, because there's more RNA that's present, we're going to want to sequence just a little bit deeper. We don't have to worry about that RNA being fully intact, because we are going to be using a different priming method for that. So this approach is more amenable to things that might have slightly reduced quality. And with this, again, we're going to be able to see everything we saw in the mRNA sequencing, but we're looking at all transcripts, not just the polyadenylated ones. So this is giving us just a little bit more of a view of what we're looking at. Now, if we're trying to decide between these two, our biggest question here is, do I care about all of these transcripts or just those that are going to turn into a protein? And that's usually going to be our deciding factor between these two approaches. Now, with either of these approaches, we're going to have the same general bioinformatics solutions. We're going to be getting into the bioinformatics solutions, what these results will look like, and how we can actually tease them apart in our part 2 webinar of this series. But as a highlight, we'll be able to get things like mapping and hit counts, gene expression, splice variants, gene ontology. We'll be able to detect things like fusion events. We can do de novo assemblies, things of this nature. Now, when we were looking at this chart before, I mentioned that we have two other approaches. On the left-hand side here, we have something called high-throughput gene expression screening. This is a modified approach to our mRNA sequencing, and it's really ideal if we have a large number of samples. So there might be times that we're going to want to do RNA sequencing as a means of screening our samples or looking at what kinds of results we have. So similar to mRNA sequencing, we're going to start with that poly A selection, looking at that poly A tail. But instead of sequencing the whole transcript, we're just going to do the tail end. That means that we're going to get a gene level view of what's present, but because we're not looking at the whole transcript, we're not getting the transcript level view. Because we're looking at just the smaller end here, we're going to get our gene expression, but because we're not looking at the full transcript, we are not going to be able to detect things like variants, alternative splicing, and so on. I want to take us into a little bit more detail about how this approach works, as it's a little bit unique and modified compared to our standard approach. So with high throughput gene expression screening, again, this is ideal for times when we're going to do a screening-based approach of 96 or 3D4 well plates. With this, we're actually going to start straight from lysate. So instead of doing a traditional RNA isolation that we're doing for our standard approaches, we're going to go straight from the lysate and do library prep directly from the lysate. This allows us to really consolidate the step and also work with smaller sample inputs. We only need about 5,000 cells for this entire workflow within each well. Again, we're going to be doing that 3' end counting. So we'll be attaching a UMI and a sample barcode to every single sample in the RT reaction. And then we'll be able to consolidate this all together into one pool for downstream library prep and sequencing. And our target amount here is going to be about 1 to 2 million reads per sample. I know it sounds like a small number when we think about the 20 or even 40 million we were looking at before, but again, because we're only looking at that 3' tail, we don't need to get as many reads because we're not covering the entire transcript. So this general approach, we would start with our experimental design and do this sample treatment, a variety of different treatments that we can use here. So you can think of things like if you're doing drug discovery or compound treatment, you can also think about things like doing CRISPR edits or testing different media solutions, responses to cells, cellular response to different sorts of screening methods. There's really a wide variety of different things we can do that we might want to use this sort of approach. Now, once we've done our treatments, we're going to lyse the samples and freeze them, and then you would send them over to Azenta. And now at Azenta, again, we'll do that library preparation, we'll do the sequencing, and then we'll give you back your data. Now, again, we'll go into this in more detail in part 2, but as a high-level view of what we would expect to see, we're going to always get back our raw data, but we'll also do mapping and hit counts, and then a count matrix transformation. That allows us to actually visualize what this gene level expression is. From there, we can do further downstream analysis, similar to what we were seeing in our standard RNA sequencing. And again, we'll go into more detail in that in part 2. Now, lastly, when we're thinking about these bulk RNA sequencing methods, our last method here is called IsoSeq. IsoSeq is using our PacBio sequencer. We're going to be doing, again, a small number of reads, but instead of looking at using a small read technology, we're using a long read technology, which means that we're taking that poly A tail again, pulling out that transcript, but now we're going to look at the full length transcript completely end to end. This is really ideal for identifying new transcripts, identifying alternate isoforms, alternate splicing, and also for doing transcriptome assembly. Again, we're thinking of this one now as looking to identify everything that's in this pool that's novel and unique. So really giving us a good idea for what each of those flavors of the fruit are. So in so much detail. Now, the way that this works is that with PacBio IsoSeq, because we're looking at long read technology, we can really overcome some of the barriers that we might encounter with our standard RNA sequencing approaches. So if you think of a full transcript or a full gene, you can have many different isoforms that are present. And you may, when we look at our Illumina sequencing, we'll be able to tell the variability of these different exons, but we won't know necessarily what's connected to what. We'll be able to, again, get this variable level of frequency of expression, but I don't know what these actually look like as their final transcript. With IsoSeq, because I'm sequencing it completely end to end, each individual molecule, I'll get to know exactly what these different isoforms look like. Again, really novel for things that are discovery-based or assembly-based, so that we can learn more about what's actually present in these samples or in these genomes. Now, moving on from our bulk RNA sequencing approaches, I want to move into single-cell sequencing. So let's go back to that fruit salad. So as we discussed, when we start out with our complex tissue or our fruit salad, we're going to have all of these different fruits that are present. Bulk RNA sequencing is taking all of that and turning it into a fruit smoothie. Single-cell genomics is actually taking apart this fruit salad and assembling all of the different fruit pieces and looking at each piece individually. What this means is that I'm going to be able to tell with a lot of detail exactly what fruits are present and how many any particular fruit is. I'll be able to tell if there is just that one apple slice that's in there or if I have a whole bunch of apples present. I also will be able to tell, if I've done enough depth here, what kinds of apples, or if I have red grapes versus green grapes, or if I've included, any sort of other novel fruits that maybe I haven't seen before. I'm going to get a really good detail for all of the things that are present within my fruit salad or within my sample. Now, caveats here, though, is that while I'll be able to get a really good view of all of these individual cells or pieces of fruit, I don't know what's touching what. So I'm not going to be able to tell if that apple is always nestled next to a banana or if the apples are sprinkled throughout the salad uniformly. Those are things that I will not be able to see with our single-cell genomics. So to do single-cell genomics at Azenta, we use our 10x Genomics platform. This offers true single-cell gene analysis. And there's actually a few different solutions that we can use here outside of just standard RNA sequencing. I'm going to go into this detail in just a moment. Now at Azenta, we actually have a lot of experience with single cell technology. We've been using this for nearly a decade. And without during that time span, huge advances at both Azenta and 10x. So this includes different protocols for actually preparing tissues and samples, as well as different types of library preparations. So not only just single cell RNA sequencing, but also ATAC and multiome. And moving ahead in the future, we're going to be offering even more solutions, especially with regards to ways to help submit your samples. Now, along with all of this, we do have a lot of experience in this space. And this includes both with the library preparation component, the upstream QC components, and even ahead of that, ways to help optimize your sample submission. And I want to go into this in a little bit more detail. So the standardized workflow for single cell sequencing is that you would start out by isolating your cells and putting them into a cryopreservation media, freezing the cells, and then shipping them. This means that you're going to need to have cells in a single cell suspension. From there, you would send this to us. We will assess the viability. I'll go into that in just a little bit more detail in a moment. Remove the dead cells, and then prepare our single cell libraries. Single cell libraries are prepared as shown here on the bottom. So we're going to have 10x barcoded beads, and then the cells and reagents coming together. All of these come together in a little blob of oil. And that means that we're going to do a unique RT reaction for every single cell. This allows us to get that true single cell analysis. So we'll be able to get all of the information for the RNA present within each individual cell that's joined together in one of these emulsion beads. From there, that library is put together and sequenced on the Illumina short read sequencer. Now, with that QC that I discussed, we can do a dead cell removal with magnetic beads to help remove the dead cells. Really important, because when we think about single cell sequencing, we need live cells in order for us to be able to process that entire workflow. So what we see is that after we do dead cell removal, we can really increase cell viability, and that gives us overall better results in terms of detection and quality scores. However, it might not be feasible for us always to get a single cell suspension or a single cell solution for any particular sample. This is particularly true if we've collected certain types of tissue specimens, and we might not be able to do that dissociation, whether it is due to lab constraints or due to where those samples are collected, for example, at a clinical site. So with this, we actually have developed a frozen tissue processing workflow. With this approach, this is feasible for human samples, but also animal specimens. You would collect that tissue and then submit that frozen tissue directly to Azenta. At Azenta, we'll prepare a nuclei suspension. So instead of trying to dissociate the individual cells, we're going to dissociate the nuclei from within the tissue. We'll filter and pellet the nuclei. We'll do an optional gradient centrifugation if needed to remove any residual material, and then we'll get a nuclei suspension. What's really excellent about this is that while the tissue samples or the cells within them may or may not be fully intact due to the freezing nature, the nuclei do a great job of protecting the nuclear RNA inside. So even if we don't have intact cells, we can still have excellent intact nuclei with great high quality RNA. This allows us to be able to proceed with samples that might not otherwise be feasible for doing a full dissociation and using the cellular RNA. And so with that, instead of doing single cell RNA sequencing, we'll do single nuclear RNA sequencing, or snRNA-seq. Now over here on the right-hand side, I'm showing you a few different figures. This is comparing fresh versus frozen tissue for that single nuclear isolation. So there are a number of tissues that are really well established as being ideal for single nuclear studies. This includes samples like brain, where we see there's a lot of additional secondary structure and connective tissue that makes it really difficult to dissociate in general. So here I'm showing you brain, liver, kidney, spleen, and colon. And what we find is that depending upon the tissue type here, we have either nearly identical isolation, or some are even improved once frozen, because it's easier for us to actually dissociate from the frozen tissue versus the fresh. Our QC results are really consistent across the board. The number of genes detected is also consistent, and the clustering, or the final net result or analysis, is also consistent. So this is a really nice alternative if we don't have a good way of submitting those samples within that single cell suspension. Now, again, what about the analysis? So I will go into more detail here regarding some of the analytics in our next workshop, but I want to give you a quick highlight here of things that we can see. So with our single cell study, one place where this really shines, again, is identifying those rare cells that we would miss otherwise, right? That single apple that was present in our fruit smoothie. So here I'm showing you an example of 4 different samples on the left-hand side. We have our untreated, our control, and then 2 different drug treatments. And we can see that the bulk of the cells here are kind of shifting upon treatments. The untreated and the control are the same. But one thing that we notice is that there's this little population of cells over here on the far right-hand side that we see in all of these. And if we overlay all 4 of these pictures together, we'll see that the untreated and vehicle-treated controls are really about the same. Our 24 and 48, we see that shift up, but we see that these resistant cells really remain the same across all of these. And then we can do a deeper dive on those resistant cells to learn more about them. I want you to think of these resistant cells as those apple slices that we might not have otherwise tasted within our fruit smoothie. Now, for this analysis, we are very pleased to offer Azenta powered by Rosalind. Rosalind is a downstream analysis software that allows us to do deep analysis. You can see I have a video here going that's just showing some of the examples of things that we can see. Rosalind is really excellent because it's very easy to use and has publication-ready figures. We're going to go into a lot more detail about the types of analysis that we can get from Rosalind and what these final figures look like in part 2 of this series. Okay, now switching gears again, I'm going to talk about digital spatial profiling. So if we go back to our fruit salad, we've talked about how we have this complex tissue with the fruit salad. We're going to look at our fruit smoothie for our bulk RNA sequencing. Single cell sequencing, we're going to sort apart all of the little pieces of fruit or these individual cells and look at them in more detail. When we do digital spatial profiling, we're going to go back to that fruit salad and we're going to actually look at the fruit salad in layers. So we'll be able to see exactly what fruit touches which, but we're only going to be able to look at it one layer at a time, which means that if I have apples and bananas on the top and then I get to the bottom and now all of the apples are maybe touching grapes, I won't be able to see that from just that one slice, but I will have a really good idea of what's happening in terms of any particular tissue slice. So I'll be able to tell what's adjacent to one another. This can have a really big impact depending upon what we're looking at, because we might find that there's specific trends with regards to where and how these different cell types are present. And so to do this, we're going to use the NanoString Genomics Digital Spatial Profiler, or DSP. I'll be referring to this as DSP for the majority of the rest of the talk. This actually allows us to do both spatial RNA and protein expression analysis. I'm going to focus primarily on the RNA side, but there are actually options here. And these are going to be targeted analyses. Now, when I say targeted, it's actually a wide variety of targets that we're looking at. It is a targeted assay, which means that it's going to be ideal for human or mouse. However, it can target the entire transcriptome depending upon the panel you choose. And I'll get into that detail in just a moment. Now, when we think of spatial genomics, why is this important? This is a really kind of a nice figure that describes what I was explaining with our fruit salad or fruit smoothie analogy. So when we start with our bulk RNA sequencing and we look at our net results, which again, we'll get into all of these in our part 2 of this series, you're going to get that overall average gene expression. With a single cell, we're sorting apart the individual cells, and so we'll get the average expression or individual expression from each cell. We'll be able to see both the individualized expressions, but we can also start to aggregate things together. But with our spatial genomics, we're going to be able to see that within that 3D context, so really preserving the cellular networks. This is really important when we think of some of our more complex tissues or sample types. This can include just, again, particularly interesting tissues, for example, brain tissue, which has a lot of different cell types happening, as well as a lot of oncology samples where we might see that we're getting a lot of infiltrating cells or different cell types or things that have really changed from their natural state. Again, as I mentioned, we also do have the ability to add in that protein expression component, which means that we're really able to kind of plex these together to get both spatial and imaging from the exact same sample. What's also nice about this is that we're going to be able to retain samples for reuse. So the digital spatial profiling has a highly multiplex IHC approach. It provides us that spatial imaging and provides also the multi-omics. Again, you can do RNA and protein or RNA or protein, your choice. It's non-destructive. And then one other thing that's really critical here is that we can use FFPE. I really did not go into a lot of detail about what we need for any particular sample type. Typically speaking, we would use fresh or FFPE for DSP. 10X typically is going to be a fresh or fresh frozen sample, but there are some caveats to that. With regards to bulk, we do have flexibility again on fresh frozen, or FFPE. But this is really ideal because depending upon the specimen you have, you can use archival samples to actually do this analysis. The actual workflow, you would first start out with the panel. These panels can be small targeted panels ranging from different types of interest to larger panels, including a cancer transcriptome atlas and the whole transcriptome atlas. Once you've selected your panel, you're going to ship us the tissue. This could be the actual tissue on a block, or it could be a series of slides. We will stain the sections and then help you determine your areas of interest or regions of interest, also called ROIs. There's a few different ways that we can do this. It's actually something that will work with you to help pick out what makes the best sense, what's the best approach here, depending upon the goals of your study. Now, once we've figured out the regions of interest, we'll be able to actually do the analysis, and then we'll be able to the lab analysis, and then we'll take those results and we'll put it together to analyze those spatial expression profiles. Again, we'll go into that in some more detail in our part 2. So again, our applications here are really wide-ranging. I mentioned oncology, I mentioned neurology, but there's other places here, including immunology, where we're looking at immune cells and immune response, as well as developmental biology, where we're looking at things that are developing over time. We also want to think about, when we're putting these studies together, that while it's one slide at a time or one tissue slice at a time, we're typically going to look at a series of samples so that we can start to do comparisons across them. So for developmental biology, as an example, we might have different time points throughout that development. Now, as I mentioned, there are both small panels and large panels. The two largest panels that are available are the Cancer Transcriptome Atlas and the Whole Transcriptome Atlas. And these are typically going to be the most common tools that we're going to use. Trying to decide between which of these you want really depends on what sort of outcome or analysis you're looking for. Whole Transcriptome Atlas, just as it describes, is a targeted panel that covers all of the genes in the genome that are protein coding. The Cancer Transcriptome Atlas is a subset of about 1,800 genes, but really ideal if you're focusing on oncology or if you're looking at panels that nanostring and you want to do a combination of panels. The Cancer Transcriptome Atlas is really very comprehensive with regards to aggregating those panels. Now, again, if you decide that you want to use this approach, everything comes with those consultative services that I mentioned earlier, so we can actually walk you through the different panels and help you decide which one would be the best. So I'd like to actually now move us beyond our RNA sequencing. So when we think about RNA sequencing, again, we're looking at everything that is going to be at the RNA level, but eventually, most of the things we're looking at, including that bulk RNA sequencing, the single cell, and the DSP, we're focusing on things that are polyadenylated, which means they're things that are going to turn into a protein. So proteomic profiling is a really nice complement to your studies. I want you to think of the different studies upstream here as ways that they can lead one to the other, one to the next. We're going to have strengths and different weaknesses or different things we'll be able to detect with any of those in our downstream analysis. But the icing on the cake or the cherry on top will be doing our proteomic analysis. This is a really great way to validate the results of what we've already seen. So for protein biomarker discovery or confirmation, we offer Olink. Olink is really ideal for looking at a subset of proteins or a panel of proteins of interest to help confirm what's going on and confirm those results. Olink is extremely sensitive. It offers a wide variety of sample types with very small volumes. So this can be as low as 1 microliter, and the sample types can be more traditional samples, but it can also be some very rare and interesting things, such as human tears, as an example. But it's highly specific using a dual antibody approach. So that dual antibody approach is actually shown up on the upper right. What you want to think of here is that you have two different antibodies that are going to attach to that protein molecule, but each of them is individually barcoded. So when antibody A attaches and antibody B attaches, the barcodes, if they're a correct match, will link together, and this dual barcoded system offers really excellent specificity for that particular protein. In the event of a mismatch or something that was not correctly able to be captured in no specificity, we're not going to see that link together. And that allows us to get a really nice clean readout of what's happening, because we'll make sure that we're not getting any non-specific binding, which is always a concern when we're working with antibody approaches. These are pre-developed assays that are available in a 48 or 96-plex configuration for human or mouse. These are the most common. However, we will soon be offering a larger panel that's about 3,000, and that will be available coming soon. Again, low input from a variety of starting materials. So the standard starting materials here will be serum, plasma, or cerebrospinal fluid, but there is actually a wide variety of tissue or of sample types that we can accept. So if you have a different sample type or you're curious if it will work, please do reach out and we'll let you know. Again, the main panels here are that 96, which is a target 96, and it's a qPCR readout, as well as the target 48. There's a few different panels that are going to be available for these, and you will pick and choose which panel makes the most sense based off of what your targets are. The larger 3,000 panel, which is coming soon, offers a wide variety of options, including a mixture of these different target 48 options. Now, the way that this will work is that you would start out with getting your samples. These, again, can be fresh samples or archival samples. And you will discuss your experimental design. What panel do I choose? And then submit your samples. Once the samples have been received, we'll be able to process it using that Olink PEA technology, where we're going to use that dual barcoded system. And with this, you'll receive downstream data analysis. We'll go into that analysis in some more detail in part 2. This also is going to come with a certificate of analysis, and we'll review that as well, as that offers some additional details with regards to how this was done and other information that we can learn from our study. Now, putting this all together, we have a number of different solutions that we discussed today. Again, today we really discussed that high-level view of how all of these work and what we're going to learn from any particular analysis. We'll be going into a lot more detail in part 2 with regards to what the analytics provide, what sort of downstream deliverables do we get. So when we're looking at these, what we'll be able to ascertain from any one study versus another, as well as where these technologies complement each other. Now, as a full review here, so with our bulk RNA sequencing approaches, this included both the Illumina and PacBio options. And for our Illumina options here, we're thinking about our standard bulk RNA sequencing, as well as our high throughput gene expression screening. These are going to be the fastest and most cost effective. We're looking at homogenized samples, again, that fruit smoothie, to get transcriptome-wide identification. We'll be able to get gene expression, and we'll be able to do this at a high throughput method if we prefer, but we, again, are not going to be able to detect small or rare cell populations, and we are not going to be able to necessarily do the same level of discovery that we might with other approaches. Single cell and single nuclear RNA sequencing allow us to profile thousands or even 10,000s of cells at a single cell resolution. Again, we're taking that fruit salad and we're breaking it apart into the individual pieces. We'll be able to get that heterogeneity that's in the sample and identify those rare cell types that we might have missed with our bulk RNA sequencing approach. I didn't discuss this today, but there are a number of other multi-omics options that we can add to it. This can include things like immunoprofiling, attack sequencing, which is going to be on the DNA, or multi-omics, which is going to be aggregating this all together. Now, this does mean that we are doing a dissociation, whether we're doing it from the single cell or single nuclear level, but that means that we lose the context of that cellular physiology. Spatial profiling allows us to keep that morphological context because we're looking at that individual slice across our fruit salad or tissue. We'll be able to do simultaneous profiling of protein and mRNA if we choose. We can select our regions of interest based off of our interests of that sample. So this can be an aggregated area or individual pockets or even single cells. And there's a number of different panels that we can choose from. So we can go for a small targeted panel, a moderate panel, the Cancer Transcriptome Atlas, or the larger whole transcriptome atlas, which is really all encompassing. Finally, we have our Olink protein biomarker method. This allows us to do our downstream analysis based off of the results that we learned from our upstream RNA sequencing. We'll be able to measure multiple proteins simultaneously, depending upon the panel that we choose, ranging from about 40 to up to 3,000 proteins at the same time. This requires very minimal input amounts. Again, we're looking at biofluids, but small inputs are fine. And this has high specificity and high sensitivity, as well as high throughput, based on that unique Olink PEA technology with a dual barcoding. Now, we know that you have a number of different service providers to choose from, so I just want to highlight why Azenta can be a great solution for you. So in discussing everything that we have today, we do offer great efficiency with less effort. We are one of the, we have the highest on-time delivery rates for next generation sequencing, and including that Olink technology, which is still considered one of our next generation sequencing offerings. We have extensive experience and excellent customer support. So we've sequenced over 200 different species, over 100 million gigabases of data, and over 100 different types of samples. Now, today we really focus specifically on our RNA sequencing technologies, but you can see here we actually offer a really wide variety of different solutions. So if you do have something else that you would ever like to discuss with us, we're happy to have one of those complimentary consultations to talk you through it in more detail and help you find the best solution for you. And with that, I'd like to thank everybody for attending, and we're happy to move on to our Q&A roundtable discussion.
Great. Thank you so much for that deep dive into transcriptomics solutions. So we did have some questions trickle in, so we're going to move to the Q&A portion, which will be facilitated by Sibi.
Thank you, Brianna. Yes, we have a few good questions in, and we're excited to answer them. Just a quick reminder, if you do have questions, please utilize that Q&A module there. Before we jump in, we also have Dr. Bennett joining us as a panelist, and since we haven't introduced you, Alan, would you like to give a brief introduction for yourself as some of the questions come in?
Sure. My name is Alan. I'm a study manager here at Azenta, so I'll be happy to answer any of our service questions you may have. My own background was basically in hematopoietic stem cell transcriptomics and then platelet transcriptomics for a postdoc before I joined Azenta.
Awesome. Well, thank you for joining us, and thank you, Dr. O'Hara, for the presentation. So, Alan, I'll direct the first question to you. It's kind of a two-parter that I feel like is something that we run into around sample quantity. So if there's low sample quantity, are we still able to do total RNA sequencing? And also around sort of low quality, if our QC shows RIN values 3 to 4 or low in that, are we still able to process that? How do we approach that? Would you mind speaking to that a little bit?
Yes, I'll just start telling you a little bit about our typical approach. Is definitely poly-A capture, which assumes basically, a high concentration and really intact RNA. So high DB200 values, high RIN scores of 6 or above. So when we receive the samples, we will always QC them, whether we do extraction or whether you submit total RNA. And once we QC those samples, we'll send those results to you out as soon as they're available. And then if we notice that a poly-A project actually is more degraded than expected or has a lower concentration, then we essentially have a couple different approaches. So for degraded, a good way to get around that is just doing ribosomal RNA depletion, which removes all the, ribosomal RNA, which is something like 99% plus of cellular RNA is ribosomal reads. You don't want to lose those in your experiment. So if we deplete those ribosomal RNAs with a probe kit, then we actually just basically end up with what would be messenger RNA enriched anyway. So that's our second approach. And then if you find that it's not only degraded, but it's perhaps degraded or it's not measurable, we actually have ultra low RNA input approaches, which essentially just have some early prior on amplification steps to deal with the fact that there's not much low input. And those projects are pretty much best effort because there's a phenomenon called dropout, where if a transcript is rather lowly expressed, as you're sampling less and less of the total pool of cell RNA, you're less likely to see it. So it's basically just like a population statistics problem. It's like if we're only sampling, 5,000 out of a million species, we might not see something that's only at 1%. So that's called dropout. So that is sort of one risk of low input, is you might not be as likely to see your favorite gene of interest. But in general, we find that it works incredibly well. The data is quality. We still quality control the data. So the bases you get will be, Q30 scores really high, really believable data. We just can't guarantee that with ultra low input that we'll see, a given gene A or a given gene B, because it's still biology. But that's sort of our approaches for degraded or low input samples, is two different library prep approaches that sort of are designed to deal with those with different chemistries.
Awesome. And Andrea, I think one of the questions that came in that maybe you can speak to a little bit more is around high throughput gene expression screening. So, what are its advantages over, say, older microarray technology? Could you speak to that a little bit?
Absolutely. So unlike a standard microarray approach, we're going to have very specific targets that we're looking at. Depending upon the array that you're looking at, it might have hundreds or even thousands of targets. Unlike the microarray approach, high throughput gene expression screening is totally unbiased, which means that we're really going to capture anything with a poly A tail. That unbiased approach allows us to get a lot more depth, a lot more variety, and see a lot of different information that we might otherwise miss with a standard microarray.
Awesome. I do have a follow-up that maybe you can kind of add on to. I know that sometimes we get the question of whether or not we consider sort of that DNA treatment step prior to sequencing. Do we sequence from cell lysate solutions, or do we have to have like RNA extracted and purified in the preparatory steps? Could you kind of add on to that with some clarification around that?
Absolutely. So with the high throughput gene expression screening protocol, we are going to be working directly from cell lysate. There is not an RNA extraction step happening here. So we are going straight from that lysate. If you decided to submit RNA, we can accept that as well. But again, there's no need to actually do that full RNA extraction. Now, that being said, there's also not a DNA pretreatment step during this protocol, but because of that very special library preparation method we're using, we're typically not going to be concerned about DNA contamination, as it's very unlikely to be impacting this as long as we have a high quality sample. So as long as we're processing this within that specified time frame for lysis preparation, we should be good to go without any additional treatment steps.
Gotcha. Awesome. And Alan, I think it was in the context of single cell RNA sequencing when this question came in, but could you speak to whether or not that technique can be used for diseased plant tissue, like leaf material?
So yes, that's not a hard no. We basically would have to default in that case to single nuclei sequencing, because the requirement, as you may remember, for a single cell is it's basically cells are being pushed through a microfluidics channel, at a really high rate of speed. The channel is very small, so any kind of dead or dying cells would cause it to clog. So we would have sort of a concern of the cell viability wouldn't be that great for the plant cells to begin with. The diseased or dying, if the virus, if it's a plant, I was thinking virus, if it's a plant pathogen, if there's a lytic cycle going on, a lot of cells are being broken open, necrotic, that would cause failure. So typically for plant tissue in general and diseased plant tissue for certain, we would just recommend that the customer extract nuclei on their end. And then, we can do a particle count to make sure that there's likely enough nuclei to load on the controller. And we really push, making sure that step is likely to work, because with 10x, the bulk of the cost is the 10x controller loading step with that microfluidics chip and reagents. So we never want someone to be, on the hook for that if it's not incredibly likely to work. So that's why we would really push sending in single nuclei ready to go for those particular studies.
Awesome. And Andrea, one of the questions that we had are sort of around that, are there any disadvantages in using that single nuclear RNA sequencing over single cell RNA sequencing? And maybe when you kind of answer that, you can also speak to sort of the minimum number of cells that are required for single cell RNA sequencing and whether or not we can use that for bacteria.
Absolutely. A bunch of great questions. So starting from the top here, single nuclear versus single cell RNA sequencing. So they're going to be similar, but they're not going to be the same. Single cell sequencing is going to be using the cytoplasmic RNA. Single nuclear RNA sequencing is going to be using that nuclear RNA. Now, depending upon the tissue type or sample type, you might find even within the existing publication record, that they're really going to heavily skew towards one or the other. Some tissue types, such as brain, are very difficult to dissociate, even on the best of days. So majority of studies are going to be in the single nuclear space. Now, that being said, there's going to be direct correlations between single nuclear and single cellular RNA sequencing. There will be some differences because, again, we're looking at the nuclear component versus the cytoplasmic, but there's definitely going to be a lot of trends and similarities. With regards to which one is necessarily better, it's really just going to depend on your approach here. I wouldn't say that any one is particularly better or worse. However, some are going to be more compatible with certain sample types. Other things to consider is that if I have a very complex tissue with a lot of fragile cells, I might actually lose those if I'm trying to do that single cellular dissociation. So single nuclear can actually give us a lot of that added data that would otherwise be hidden. Think again back to that case study that we discussed where you might not be able to see everything if you were losing those cells from that freezing thawing process. Regarding the minimum number of cells, in a perfect world, if we could start with the number of cells that we would want to target, which is up to 10,000. However, we really recommend submitting more than that. So a minimum sample count would be 50,000 cells per sample, but the ideal amount is going to be almost tenfold that, 500,000 or more. If you submit that higher level of cells, that'll allow us to do that whole protocol that I discussed, including the dead cell removal, to really improve the quality of the sample. Now, the last question that we had was, single cell RNA sequencing for bacteria. Yeah. So with the single cell sequencing, we do need it to be a polyadenylated sample. So bacteria would not be compatible for this workflow because we are going to be using that poly-A tail to capture it. So any type of animal sample, again, Alan mentioned plants, if you isolate the nucleus, but we do need them to be polyadenylated transcripts. Non-polyadenylated transcripts are not compatible with this workflow.
Awesome. And I think the context, we kind of lost the context of this question, but the question is, can this technique be used with transgenic Arabidopsis leaf tissue? I don't know if we can, Alan, if you can speak to, what services we can use.
I think that was in the context of a high throughput gene expression, because that was the last service we talked about, or maybe the proteomics O-link. Gotcha. I'm not sure for either of those. Andrea, are you sure if there's a?
You know what? Why don't I go ahead and take this one, Alan? Yeah. So let's just go top to bottom what we can and cannot do with these different sample types. So starting with standard RNA sequencing, that one's going to be compatible for plant or animal, no problems there. High throughput gene expression screening, we're going to want to use that for typically cell lines. Plant samples would be really difficult for this because of that cell wall. I'd be worried that we would get incomplete lysis. That being said, if you were able to get somehow a single cell suspension of plant cells, which seems very atypical, we might be able to see what we'd be able to do. But again, typically that high throughput is really meant for doing cells in suspension and animal cells at that. Regarding single cell, again, for plant samples, you'd have to submit already isolated nuclei. We can't do that nuclear isolation for you. But if you had isolated nuclei, we'd be able to do the single nuclear RNA sequencing. Last step would be the O-link technology. O-link is specifically for human and mouse only, as these are targeted for very specific proteins.
Awesome. And Andrea, another one for you regarding sort of microRNA sequencing. Can you clarify the samples, like requirements, and if one needs to look at cell-free plasma or the RNA quality yield is not optimal often?
Great question. So for small RNA sequencing or microRNA sequencing, we can accept two main different types. We can accept a variety of different sample types for RNA extraction. We can do total RNA extraction or targeted purified small RNA extraction. Now, depending upon the type of sample that we're looking at, we may want to opt for doing that very targeted purified small RNA approach, particularly if you're looking for cell-free RNA. We know that these small RNAs are typically circulating at a higher volume. You are correct that the overall total RNA quality may be poor because the larger RNAs, including ribosomal RNAs, will be degraded, which are typical markers for this. So it will bypass some of that standard QC with regards to the quality control. But again, the small RNAs may still be intact. So if you're thinking about a very specific sample type, I do recommend following up with us later and we can discuss, as we have a number of different options within our toolkit to help extract those particularly precious samples, including those small RNAs.
Awesome. And I'd like to open this up. Again, this is more of a clarification around single cell extraction protocol that someone wants some clarification for. So the question is, would our in-house single cell extraction protocol work for plants as we've described?
No, our in-house protocol is pretty much for cryopreserved tissue. It doesn't even work particularly well with frozen mammalian tissue, so we're not confident that it will work with plants. We could potentially pilot it for you, but ideal would just be send in the nuclei, because 10x, like, we want you to avoid the actual major cost of the project, which is failed loading. So that's why we're really insistent on sample quality at the get-go. So ideally, you would just extract nuclei, count them to make sure that you're not wasting your time sending in samples that aren't high enough for particle counts, and then we would just load them on the controller for nuclei.
Awesome. I'd also like to ask a question around sort of the 10x Genomics workflow. Could you, Andrea, speak to sort of how the workflow differs for methanol fixed cells?
Great question. So when we're submitting samples for 10x, there's actually a variety of different ways that we can submit samples. So the standard or most traditional method would be using cryopreserved cells. This is going to be in a special buffer that keeps the cells alive. You want to think of this as a similar buffer to if we were going to freeze down cells in suspension so that we could thaw them and grow them out again in a new flask. That's going to be our most standardized approach. However, if I have cells that are particularly fragile that won't do well with that freeze-thaw approach, I can actually do everything to get these samples to be their most robust population before I do that fixation approach, and then actually do a methanol fixation. The biggest caveat here is that once that methanol fixation is done, there's not a lot of additional QC that we can do upon receipt. So you really want to make sure that you have high quality, very pure, high viability cells prior to doing that methanol fixation. Alternatively, we can talk about those frozen tissues. Again, with those ones, we prefer to do the nuclear isolation. This is going to be a totally different approach altogether, where we're really focusing just on those nuclei. And with those types of samples, we can either accept the frozen tissue itself or the already extracted nuclei. So you really have a lot of options at your disposal, depending upon what makes the most sense for the samples that you have, as well as how you've collected them and what you're able to actually process on site.
Awesome. And Alan, a question that we have, one of our attendees has samples that are human cells infected with a known RNA virus. Will they be able to examine both the human and viral gene expression levels with our sequencing, and how many reads should they aim to achieve in that context?
Yeah, sure. So you could definitely examine both human and viral gene expression. If the virus is expected to be polyadenylated, we would just capture it with poly-A capture. If it's not polyadenylated, we could think about RNA removal, which should then leave, viral message plus cell message. We would ask, about the BSL level. If it's like a BSL 2+ or 3 virus, we would typically ask for you to do some work at the end for extraction to render it inert before you send it out to us. For really dangerous things, we often ask for like infectivity assays to show that they're confirmed to not be infectious. So, we can help with even dangerous viruses with caveats. And then how many reads you should aim to achieve is really dependent on sort of the frequency of the virus in the cell, which is a little bit, needs to be empirically determined. Like if you know that there's 50 viral copies for every 10,000, human copies, like we could totally back calculate the math for that. I will say that in general, NGS is so deep that it's not typically a problem. If you're sequencing on a platform like a high-seq or something, it's 350 million reads. So depending on sample number, we would basically, the answer to this is like, if you know the frequency of the virus, we're happy to help you determine the number of reads that you would be likely to need to capture it. Other than that, if it's not known, it's a new pilot project, it's always empirically determined. Like you might just have to do a study and find out sort of what level of virus is being expressed in these cells. And maybe that's part of your study. If you have a highly virus that's like really highly proliferative and you're having some experimental therapeutic to knock that down, like maybe you just want to show that there's less reads in a treatment. So often that's what you're actually looking for is a decrease in viral reads out of the background of human reads or mouse. So
Awesome.
So yeah, we can definitely do that. We're happy to discuss any project with you. Just virus is a little weird because we want to know if it's polyadenylated or RNA depletion, but we're happy to walk you through sort of the best approach for any given virus, any given cell.
Awesome. And we have time for one more question, and Andrea will kind of end it with you. A question from the attendee was around, are we considering widening our samples to other vertebrate samples like zebrafish? Could you speak to that?
Sure. For which technology does it specify?
No, it does not.
Okay. Let me go ahead. So again, when we're thinking about our different technologies, some of these are going to be really specific for the types of platforms we're using. So for total RNA sequencing, anything and everything. That it does include bacteria. We have approaches that can work for really any kind of RNA, polyadenylated, non-polyadenylated. We have options across the board. For high throughput, we're really going to be focusing on things that are going to be easily lysed. So typically thinking about things that are going to be cells in suspension, and also high volume of samples. Single cell is really going to be specific for anything polyadenylated, but that does include really any animal species that we can consider, including zebrafish. Olink is very targeted towards mouse and human because these are going to be antibody based. So they're going to be targeted for very specific proteins. With this approach, that one really is going to be locked in for these specific types. If you're interested in some other proteomics, though, please do let us know because we may have additional options. However, Olink is really going to be specific for mouse and human samples.
Awesome. Thank you. Thank you again, Alan, Andrea, and Bree. I'd like to hand it back to you.
Yeah, great. Thanks to everyone for submitting those questions. They were really great questions. And thank you to Dr. O'Hara and Alan for joining us for the Q&A session. And thank you to all of the attendees for joining us today. So the recording will be made available to you for on-demand viewing after the Part 2 airing. So have a great day, and we'll see you in a few weeks for Part 2.
Bye, everyone.
Bye-bye. Thank you.